The next speaker is Corey Watson. So Corey Watson is an assistant professor in the Department of Biochemistry and Molecular Genetics at the University of Louisville School of Medicine. And he's going to introduce his work on germline characterization in adaptive immune receptor genes. It's all right, perfect. So please, your microphone is turned off. Correct. Yeah, you have to turn on your microphone. Yes.
Sorry, I'm on my phone. Can you hear me now?
Yes, perfect.
Okay, perfect. First, just want to say thanks for the opportunity to share work and ideas with all of you today. So my group's interested in characterizing germline variation and adaptive immune receptor genes, including immunoglobulins and T-cell receptors. Today, I'll mostly sort of give examples in immunoglobulins, but for the most part, everything I'm saying is also, to some extent, at least true in T-cell receptor genes as well. So a lot of what my lab focuses on is understanding what germline variation is doing in the immune response. But because of that, I've been pretty involved in a lot of what the airr community has been thinking about and doing in this general space. So I'm going to talk a little bit about, in the first part, a little bit about what the airr-c has been doing in the context of germline variation, giving you kind of an overview of what we've been learning over the past few years and a little bit of information on what the airr-c is doing. And then I'll sort of show you some work at the end from my own lab, which is one reason that I think we should care about germline variation and immune response. So as I just said, the first part, I'll give you a little bit of background on variation, what we're learning in human and other species, and then I'll talk about some tool development and some biology from my group. So as you probably already knew or have gathered from many of the talks today, when we're thinking about germline variation, immunoglobulins and T-cell receptors, we're often thinking about how this influences or what role this has in understanding the way VDJ works and the way airr repertoires evolve in a response. So a critical component in this whole story, of course, are the genes that encode antibodies and T-cell receptors. These are, of course, starting points for VDJ recombination. And so with this in mind, and given that a lot of the analysis that many of us engage in is centered on sort of characterizing these sequences in a repertoire, an important aspect of many of these analysis pipelines are the use of germline databases. And germline databases are essentially catalogs of genes and alleles, which I'll sort of get into more detail here momentarily, in which we can take airr-seq data reads and find assignments to genes and alleles in a given species or sample. Once we've done that, we can then go on and do many of the things shown here on the right-hand side of the plot, where we get to sort of learn about features of the repertoire, signatures and repertoires that may or may not be important in disease or other clinical phenotypes. And so just to sort of step back for a second and give you a little bit more context on what I'm talking about. So it's kind of good to start by thinking about what are the genes that encode these receptors. And so I'm going to use the example to begin with the human IgH heavy chain locus. So shown here is kind of a cartoon depiction of a haplotype at the end of chromosome 14, which encodes D segments shown here in blue and J segments in yellow, sitting adjacent to constant region isotype genes. And so when we think about making databases and cataloging variation, really what we mean is we're cataloging variation that we see showing up in these gene segments. So this is the heavy chain. I'll get into more detail here momentarily. And then the light chain loci, lambda and kappa, look very similar. Won't really talk too much in detail about these loci today, but in structure at the genomic level, they look very similar. And again, T-cell receptors also have a very similar look at the genomic level, where you sort of see these expanded gene families V, D, and J segments. So again, using the heavy chain as an example, in human, what we've known for a while now, we're really just now starting to learn a lot more about, is that within each of these V segments, and this is also true to some extent for D and J segments, there's a lot of allelic diversity in the population. So this box is showing alleles that are cataloged in IMGP, which is kind of the historical primary database that's been used for cataloging variants in these genes for the last several decades. So you can see here along the x-axis are known functional variable gene segments, and then the y-axis is showing the count of known alleles, shown as black bars, and the number of those alleles that are representative of non-synonymous positions, or rather the number of non-synonymous positions found amongst those alleles are shown. And you can see that it varies between gene and it varies between phylogenetic subgroup or subfamily, where some genes have one allele, other genes have greater than 15 alleles. And an important observation that we're sort of now understanding is that we're probably just kind of scratching the surface of the extent of diversity that's out in the human population. This largely is because historically we primarily sequenced from Caucasian populations, and so now there's many groups kind of stepping into non-Caucasian ancestral groups, and we're seeing that there's just a lot of variation out there across the globe, which is interesting. And so one of the reasons that we are kind of behind in our descriptions of these loci relative to, say, other immune loci like HLA or KIR is that these regions historically have been very technically difficult to study. And one of the reasons for this largely is that they're really complex at the genomic level. So I sort of showed you this cartoon a minute ago, which was depicting mostly functional segments in the human IGH locus. But when you actually add in pseudogenes and open reading frame genes, what we see is that you get a much more complex picture of the locus, where instead of having 40 to 50 some odd functional segments, we now have upwards of 130 these variable gene segments that sit in the locus, even though many of them are non-functional or pseudogenes. But what this creates is this very repetitive locus that's very hard to interrogate with standard high throughput sequencing and array-based strategies. So what we know is that the locus, greater than 50% of the locus, is basically in a repeater segment duplication, which makes it hard to study. We also know that between people there are structural variants or copy number variants, which are shown here as n sets along the primary haplotype, where you have these large insertions and deletions of genes depending on a given chromosome. And historically, these have not been well represented in genomic databases. And so, again, these standard high throughput approaches have really been met with challenges. So when you take this allele diversity and structural complexity and put it together in people, you basically see that most of us are walking around with different haplotypes, characterized by different alleles and different structural variants. So this is data from 12 different individuals. Excuse me, not 12, I can't count. 9 different individuals in which we can see haplotypes arrayed out here as these tiles, and each tile is colored by the allele identified. And the take-home message really is that there's no 2 haplotypes in this figure across these samples that are the same. So there's basically just really extensive diversity among individuals. And so one of the sort of critical questions right now that many of us in this space are trying to understand and figure out is how extensive is this variation across populations, and how much allele sharing or how much differences can we observe when we compare, say, Caucasians to Africans or Asian groups, so on and so forth. I think this is going to be a really exciting area to get into as we uncover more data. So that's human, and that's really a very short summary in human. There's a lot more to be said about it, but just for the sake of time, I'll move on. We also know that in these many other biomedically relevant species, that the story is very similar. So this is data that was generated by Andrew Collins' group a few years ago, in which they characterize differences between C57 black 6 and FOCiS, looking at V gene variation, again, focusing on germline variants. And what you can see here in this Venn diagram is that when you look at the V gene set of FOCiS compared to C57 black 6, there's very little overlap between the V genes observed in both of those two strains of mice. So this was a bit of a surprise at the time. And we're now sort of, several of us are now trying to understand this to a greater degree and across various mouse strains. And what we're seeing is that, in fact, there is a lot more variation than we previously anticipated. So this is just an upset plot showing comparisons across 7 different strains. Again, FOCiS and C57 black 6 are among those. And what we can see when you look at the V gene sets among these strains, that the majority of sequences are unique to only a single strain. So there's not a lot of shared sequences amongst different mouse strains. And at the time when we did this sequencing, one of the sort of big observations was that when we think about databases, the majority of sequences that are found in commonly used databases did not represent the sequences we were seeing across these mouse strains. So it's sort of highlighting this sort of technical problem. And again, this is a lot where the airr community is coming in now. We can also see when we look at FOCiS and black C57 black 6 at the genomic level, so actually comparing the sequences of their haplotypes, these strains look very, very different. And so this is something that we're all trying to kind of get our heads around now and understand how this affects the way that we catalog variants in these strains. And of course, that extends into how we ask biological questions potentially related to immune phenotypes or antibody-mediated phenotypes in these different strains. And we also see in another biomedically relevant species, rhesus macaque, that the story is also very similar. And so the genomic resources for IG, loci, and rhesus have also kind of been stunted, again, for many of the same technical reasons I raised earlier. But we're now starting to see some assemblies come on the scene. And again, we're seeing variation between these reference genomes, showing that there's both allelic variation as well as structural variation between animals. And this is just data recently published by the Hedison lab, where they sequenced airr-seq repertoires for around 45 animals. And again, much like the mouse example I just gave, there's extensive numbers of alleles that are not currently cataloged in the databases we all use. And there's extensive variation between individuals. And so with the sort of human mouse and macaque example, this has really driven a lot of the work being done by the airr community in this respect. And so several years ago, the Germline Database working group was established to sort of begin addressing these problems and try to make improvements in this area. This group is currently co-chaired by Andrew Collins and myself. There's the link for it if there's anyone interested. And it's composed of many different people across the field who are interested in these problems. One of the main, our main mission is to promote the comprehensive and accurate identification, description, classification, annotation, curation, and consistent use of germline, IG, and TCR alleles across species, strains, and populations. And this kind of comes in many different flavors, but mainly it's about really thinking about how we improve databases for these genes across species. One of the biggest sort of accomplishments we've had so far was setting up this inferred allele review committee. And there's a paper here I put at the bottom which sort of outlines this sort of concept behind this committee and what it's trying to do. So it's been really focused on trying to catalog and curate alleles from airr sequencing data, and is now thinking about how this is going to extend into other types of genomic data as well, in which we know that there are lots of variants to be cataloged. And we've also begun thinking about how to build better databases and augment existing databases like IMGP through integration of different data types and different types of reporting formats and data sharing schemas. So two of the main databases that the airr-c GLDB group has been involved in is OverDB and mediated. Those are shown here. And the idea here is that we're just trying to build kind of more comprehensive database for databases for sharing these genes. So again, if there's anyone interested in these activities, you can find us usually through a quick Google search, just our community Germline Database working group, and the links down there, or reach out directly to Andrew. Okay, so that's sort of a Germline Database overview. I'm going to quickly, in the last few minutes, to sort of switch to some stuff that my group's thinking about. So my group's very interested in how does germline variation actually influence repertoire. And as I said before, this has been a sort of challenging question to address historically, just because these loci have been very difficult to study. So my group's been really focused on trying to develop higher throughput sequencing methods to basically effectively genotype dehaplotype variation in humans. And our goal here is to really understand how that variation influences the repertoire, with the idea in mind that if we can understand those initial links, that we can then go on to understand how the antibody-mediated immunity influences disease and clinical phenotypes. And so the idea is basically that we're thinking about right now, a lot of stuff we're doing a lot of centered around how does haplotype diversity sort of feed into the mechanisms that drive variation in DDJ recombination. And there's precedent for this because we know that when you look at repertoires of monozygotic twins compared to unrelated individuals, with certain features of the repertoire, in particular gene usage variation, we can see that monozygotic twins look more similar than unrelated individuals. And so, as I said, we developed high throughput approaches that leverage packed file sequencing, long read sequencing to try to overcome some of the shortcomings of short read data. I won't go into too much detail on this today, but this is a targeted method where we basically can enrich sequencing libraries for IG loci and then sequence these. And we developed a bioinformatics tool that basically allows us to go in and create haplotype-specific assemblies and then translate these into variant call sets, curated alleles and genes and structural brands and whatnot. And the idea here is that we can then take these and apply them to any sort of clinical cohort of interest and basically produce genotype call sets for the whole of the IgH locus. And so for this little snippet of data I'm going to show you in the next few minutes, our goal is really to sort of try to expand our sequencing efforts to the population level and understand these initial connections to the antibody repertoire. And so we've largely been following an eQTL type framework where we're asking effectively, do variants influence usage and other signatures? I'll sort of show you examples of effects on usage today. So do things like variants in or outside of the genes or structural variants that remove or change the copy number of genes influence the frequency at which you see those genes in the repertoire. And so I'll show you some pilot data today from around 110 people or 111 people. These are all healthy donors where we've taken repertoires from PBMCs and we've genotyped all of them using our high throughput PacBio method. So our call sets have around 6,400 SNPs, and we're cataloging usage of these sequences in repertoire. So at a high level, what we see when we ask, do SNPs correlate with usage in the repertoire? The majority of IgHV genes have at least one variant in the locus that's associated with its usage at a relatively high significance cutoff. And we see that in many cases, genes are associated with many SNPs, sometimes hundreds of SNPs, which are representative of these kind of large haplotype effects. So this is just an example to give you an idea of what we're seeing. So this is for the gene 366. This is a Manhattan plot in which plotted along the x-axis are the positions of all the SNPs that we've genotyped across these 100 individuals. The y-axis is showing the minus longitude p-value of the association. So every SNP associated with the usage of 366. So the higher it is on the y-axis, the more significantly associated it is. So this is kind of what these data look like. So all those shown in red are now shown down here, the positions on the genome relative to genes. So there are around 300 shown here. And if you look at the lead SNP, the two lead SNPs in this analysis, they are both located relatively close to one another. One's in a non-coding position and one's in a coding position. But you can see in the AA and GG genotypes on the left of these box plots that there's virtually no usage of 366 in those individuals. But as you add the sort of altering alleles, you can see the usage sort of creep up. And we see other examples where SNPs in non-coding regions, such as recombination signal sequences, as well as 5' UTR sequences, also show similar effects on usage. And then we can also see these effects of structural variants. So this is an example of the gene 438-2, which we know is in a structural variant, in which some individuals don't carry the gene in the genome. And again, we expect that those individuals will not use the gene. That's indeed what we see. But this gene is actually interesting because in addition to the structural variant, we see that there are other SNPs in the locus which are actually many hundreds of kb away from each other that explain additional variation in usage. And so this is just one example of these sort of complex effects that we see across the locus, likely indicating that the genomic regulatory elements influencing VHA recombination are probably somewhat complicated for many genes. And then I'll leave you with sort of one final point, which is that the data I just showed you is from IgM, but we actually see that many of these patterns carry through into the IgG compartment as well. So this is just an example of two genes where you can take SNPs that are associated with usage in IgM, and you can basically see that effect is also there in IgG. And indeed, when you look at the correlations of this usage between the IgM and the IgG repertoire, you see they're highly correlated. And this is effectively true for about 30 genes in this data set, at least, between IgM and IgG repertoires. And so with that, I'll conclude. So in the first part, I hope I sort of gave you an impression that immune receptor loci are both complex and polymorphic, and that there's good motivation for us doing a good job of cataloging and establishing resources for sharing these data sets. One, if nothing else, to make sure that we were able to do accurate analyses of airr-seq data sets and really learn things about the repertoire. But then also, this is important for being able to explore the role of this variation in antibody and T-cell receptor mediated immunity. The airr-seq has really taken an active role in the last 5 years in trying to make sure that we're keeping up with the times and pushing the community forward and generating these resources. If there's anyone interested, please reach out to either Andrew or I to join these efforts. And then I hopefully sparked your interest and showed you some data that might convince you that germline variation could be important in studying repertoire. And of course, for us and what my lab's doing, this is a sort of key first step in a lot of the biology we're thinking about and understanding how VHA recombination works at the genomic level. And in addition to all the airr-seq people that I would have to thank, which would be a very long list, these are folks in my lab and our collaborators who contribute to the second part of data that I showed you. And with that, I'm happy to take questions. Thanks for your attention.
Thank you, Corey. Thank you for this impressive work. I think so. I think Lindsay has a question, and probably after Victor.
That was great, Corey. Thank you so much. Two quick questions. Maybe the answers are obvious, but first, do you tend to see that when you discover new SNPs, do they tend to be in CDR3 or in CDRs or in framework or just anywhere? And then second question, does this change what we think we know about the targeting of somatic hypermutation?
Yeah, so your first question, so we see that there's pretty much extensive variation all over the locus. I wouldn't say at all that it's particularly enriched in the gene segments. We haven't formally tested that, but yeah, I wouldn't say that that's necessarily the case. And a given individual just across IGH, which is about a megabase long, we'll see anywhere from 2,000 to 4,000 SNPs in a sample. So there's just a lot of variation in IGH. In fact, we now know that it's elevated over the genome average, so it tends to be 2 to 4 times higher than what you see in the rest of the genome in terms of just SNP density. And then your second question was, does this change the way that we think about somatic hypermutation and such variation? I think the answer to that is yes. So this is something that we've actually worked on a little bit with Andrew Collins. And you can see evidence where sort of your germline alleles do have these effects on the trajectory of SHM, particularly when SNPs are sitting adjacent to hotspots. But we haven't really formalized that. It's something that we're trying to kind of get together now. Does that answer your question, Lindsay? Sorry, a bit rambly.
Yeah, thank you so much.
I have one question, Corey. Very nice talk. Regarding the dependencies you saw between SNPs and IGH usage, to what extent do you think does this expand or restrict potential repertoire diversity compared to what we thought before, the baseline, you know, not thinking about polymorphisms, not thinking about those dependencies? Do you think it actually restricts potential diversity of an individual in the VDJ recombination process, or do you think it expands our potential space that we can explore?
I think it's probably both, and I think that's sort of context dependent. I don't think we have enough data to say at the moment whether it constrains the repertoire as a whole, or whether there's examples in which that's true. We know that there are examples where there are individuals who carry these large deletions, where, you know, sometimes up to 20 genes will be deleted. We haven't really put formal bounds on that, but I suspect that in those cases, those individuals have repertoires that are somewhat constricted. But what I do think is true is that when you're thinking about individual gene segments, these SNPs can have really drastic effects. So, you know, we know that in many cases, these EQTLs basically are telling you whether an individual has that gene available in the repertoire, period. So it's not there in the naive repertoire. You're not going to select it out. It's just not there. So I think if in cases where, you know, where you're sort of trying to elicit very specific immune responses, or you're trying to elicit particular antibodies derived from certain germline alleles, I think that's where this type of data is really relevant, because if you don't have it or it's not available in repertoire, then that's a hard ask. And then, did you ask something else? Sorry.
No, that was all.
That's covered.
Yeah.
Okay.
Thanks.
Cool. Thanks, Victor.
So there's 2 additional questions, but because we are a bit late, I propose that we will take them during the last session of the Q&A. Okay.
Sure.